Remember that we said that we have some granules at the surface of the membrane? So this is where the cortical reaction is going to happen. So this is more of a mechanical block that becomes effective about a minute after the sperm egg bind together, the first sperm, okay? So if we look around that surface of the egg, all around, and we want to see that in the next slide or so, we have about 15,000 cortical granules all around that membrane, okay? So once the sperm enters, so we have here plenty of sperm looking for the lock. Remember, we say that this is a system of key and lock. So it's looking for whether they are the key to open the lock. In here, in B, this is what we see, that point in there is exactly the point of entrance of that sperm. And how we know that? Because it's exactly the point at which the cortical reaction has started, okay? So once the sperm put the key inside that lock, all these cortical granules start going exocytosis, okay? Which in this case will be a very similar reaction at what we saw happening on the acrosomal part of the sperm in the vesicle, okay? So what's going to happen is that there's going to be a lot of calcium that start propagating and creating a gradient. And that gradient gonna start from the point where the sperm start all the way up down to the opposite end of the sperm entrance, okay? So where this calcium comes from the endoplasmic reticulum of the egg, okay? So we know that the endoplasmic reticulum contains a lot of calcium and it will come from it, okay? So the content of the cortical granules are released into the space between the cell membrane and the vitelline envelope. Remember that these two are tied together with some connections of actin, right? So, and the granule is going to come at that point, okay, between the cell membrane and the vitelline envelope. So these granules contain many other things, as we can see in here. So we have proteases. The function of these proteases is to dissolve the protein that connects the vitelline envelope with the cell membrane. So this is the main function of these proteases. So remember, these want to have binding receptors that are clipped between the cell membrane and the vitelline envelope right here, okay? And then we're going to have a separation of the two membranes, which means that that lock in the surface, because remember that we have many locks, not everywhere, but we have many locks in that surface of the membrane. But because now with the release of the proteases, we are cleaving those links, now the lock is lost. So we don't have anymore. Those locks get invalidated. They can't be functional. We're going to secrete other things like mucopolysaccharides, as we see in here. These mucopolysaccharides produce an osmotic gradient that causes water to rush in the space between the cell membrane and the vitelline membrane or envelope, okay? So, and this is what happens here. Oops, with that, I just wanted to change my point to illustrate. So you see how here these spaces start being created between the cell envelope and the membrane, vitelline membrane. As we go into the next one, we see that that space becomes greater, right? Now it's from here to here. And that is because the water gets in between. So the cell becomes like swollen. And as the envelope becomes swollen, then it just takes the name of fertilization envelope. So the vitelline envelope is no longer called vitelline envelope. It will be called fertilization envelope because only is seen when the egg has been fertilized. Okay. These granules also have peroxidases. The peroxidase enzymes harden the fertilization envelope by doing some cross-links of tyrosine residues to the adjacent proteins. So now not only the logs are not functional, but they get blocked by these peroxidases, okay? Because now that vitelline membrane is so hard that they can't penetrate. They don't have mini drills to do so, okay? There is also some iolin proteins into those cortical granules, and this will form a coating around the egg. So this is what we see in here. You see? Now we have a little bit of a coating that goes around that egg. And this will be really important because the egg stem microvilli that we had at the beginning gonna support the blastomer formation during cleavage. Okay, so when I see that is not forming here, but in here, so all around we want to have these iolin proteins to support the blastomers, to keep them together. Is this process and the functions of the cortical granules understood? Okay. So now we're going to see a little video that is going to show that cortical reaction. So this is the egg. There's so sperms are going to be around, and then we're going to see what happens in real time. You see how it starts swelling, water rushes in, and it's very, very quickly, right? I'm going to see it again. So now this is the fertilization envelope, and this is the egg membrane, okay, over here. So we're going to just see it again, just so we know. That was the point of entrance of the sperm because it went all the way around to the opposite end. One more time. All around, cortical reaction, all around. Isn't that cool? I always find that so cool. Okay. Now, so this allows us to review a little bit all the different steps that I just mentioned prior. So this is the sperm. We have the tail, we have the mid-piece with the mitochondria, we have the centriole from which the tail, the flagellum originates. This is the nucleus, this is the acrosomal reaction that it had, okay? And here we have the penetration. So here, this is the jelly coat, okay? And here is the vitelline envelope. Now we have here all the microvilli under which we find the cortical granules. Because it got in touch as recognition of the key and the lock, now we will have this point having the cortical reaction. And now mucopolysaccharides, water goes in, right? Start the reaction. Look at this cell membrane, how it starts separating from the vitelline envelope. Okay, this is the membrane and this is the vitelline envelope. And now that we have that space in between, it becomes the fertilization envelope. Water continues to go inside until we have all this hard layer here and no more of the sperm are able to attach, okay? So we think of fertilization as only a means to unite two haploid cells, but it's an equally important role in initiating the process that begins development. Just because of the fact that a sperm got into that egg doesn't mean that it will become a zygote. The egg needs to get out of the arrest stage it was, right? Remember? And need to start generating all the machinery that is required for the formation of the embryo. So these events happen in the cytoplasm without the involvement of the nuclei. Okay, so the mature Urchin egg is metabolically sluggish. It's being a cell that only is being activated by the sperm with that cortical reaction and the entrance of water and the calcium wave. So the sperm activation is a stimulus that sets into action the pre-programmed set of metabolic events that are required for the formation of the embryo. So the response of the egg to the sperm can be divided into early responses that happen within seconds of the cortical granule exocytosis. And we also have a late response. And this late response takes place several minutes after the fertilization had begun. So in this image, what we have is what happens with that calcium wave that we saw a couple of minutes ago in the video. Okay, so how calcium is released depends on the species because it doesn't happen the same way across the species. So the first mechanism that I want to show is the one in B. It's not the one in A, but the one in B. Is observed in sea urchin. So I'm talking about this one first because we're talking about sea urchin. Okay, so when the sperm binds to the receptor, that is this green here on the cell surface, there is a change in conformation. Okay, it's the same type of thing that we saw when we were discussing the transcription factors, remember? So this receptor suffers as change in conformation that initiates the reaction within the cytoplasm of the egg. Okay, so we have tyrosine kinase family and PCL, and then pyruvate gets in here, and we stimulate the calcium channels. And this one, normally in this case, the IP3 acts as a secondary messenger to open the calcium channels. Okay. In A, that we have in here, is what we see in mammals. We're going to see this later, but look at the sperm of mammals. Never comes in perpendicular position to the membrane. It goes into the side. Okay, it's more in the equatorial section where it has the type of key to trigger the receptor in the membrane. So there is a subtle factor, a soluble factor, of the sperm that is introduced in the egg at the time of the cell fission. And that one is put into the PLC, okay, the phospholipase C. Okay, so that's the PLC in here. And this one is the one that activates the egg by changing the ionic composition of the cytoplasm. Remember, this is in mammals, okay? So in a similar manner as it happened in the sea urchin. So this phospholipase C splits the IP2 inositol 4,5-diphosphatase, okay, the phosphate, and it's transformed into the IP3, right? So the inositol 1,4,5-3-phosphate. But also it will activate this DAJ, that is the deacylglycerol. And those, the AP3, as in here I say, it will act as a secondary messenger to release the calcium. And the calcium is the one that causes the reaction. Now, we don't know necessarily whether this IP3 is the same that activates in mammals. We don't know yet too much. We suppose that's what we have an interrogation point in here, okay? This deacylglycerol is the one that activates the membrane protein that allows sodium ions to go inside and change the voltage, okay? So we still have very similar parent more known in sea urchin than we know in mammals, but this is the same reaction, PLC, that activates the phosphoinositol phosphate and opening of the calcium channels for the reaction. The early response of the egg to the presence of the sperm is the activation of all eggs, and it seems to be all related to these calcium ions. Without calcium, we can't activate that egg. So the same release of calcium ions is responsible for the cortical granule reaction that we saw, and this is what we have seen in this image. This is the wave of calcium. So this is the egg unfertilized. A fraction of a second later, we see here in white. Where is it? Here. Oh, no. Oh, the other one. It doesn't show what I want. So you see here this little piece of white. That's the calcium reaction happening. And we see how through time it starts going and it's bigger and bigger until it gets into the opposite end of the entrance of the egg, of the sperm, okay? So in some species, the calcium ions enter from outside the egg. In others, the ions are released from the egg's endoplasmic reticulum, as I explained it for the sea urchin, okay? In either case, whether this calcium is coming from outside the environment or from the endoplasmic reticulum, it goes across the start from the point of entrance to the opposite end of the egg. Another thing that happens early in here is the activation of NAD kinase, okay? This NAD converts NAD in NADP. And NADP allows lipid metabolism, which is required for the formation of the membranes of the different blastomeres that are going to form later on. So if we don't have a lipid metabolism, we won't be able to form the membrane of each of the blastomeres, okay? So synthesis is, membrane synthesis is critical for cleavage. And also, this one will activate oxygen consumption, which will provide a respiratory burst to that egg. So now we want to see a little bit of a video seeing this. Where is my mouse here? Okay. Oh, I need to share my computer sound.
Is visual.
What happened?
Fertilization is visualized here simultaneously by phase contrast microscopy and by fluorescence microscopy. The egg contains a fluorescent dye that becomes brighter in the presence of calcium ions. When a sperm cell fuses with the egg, the fluorescence image shows a wave of calcium ions that sweeps through the cytosol, starting from the initial point of sperm-egg fusion. Following the path of the calcium wave, we see a membrane called the fertilization envelope rising from the cell surface. The fertilization envelope protects the fertilized egg from the outside environment and prevents the entry of additional sperm. The rise in cytosolic calcium triggers an elevation of the fertilization envelope through the process of exocytosis. Exocytosis releases hydrolytic enzymes stored in vesicles. Action of the released hydrolases causes a swelling of material surrounding the cell, which in turn elevates the fertilization envelope. Exocytosis can be visualized directly in this system. For this purpose, the plasma membrane is labeled with the fluorescent dye, seen on the right. Each time a vesicle fuses, it leaves a depression in the plasma membrane, which, in the optical sections shown, appears as a ring of increased fluorescent staining. On the left, a differential interference contrast microscopy is used to directly view the exocytic vesicles that underlie the plasma membrane. The vesicles are visible here because they are densely packed with protein and consequently have a different refractive index from the surrounding material. Each time a vesicle exocytoses, it disperses its contents and disappears from the image. This effect is best seen when we step back and forth between adjacent frames of the movie.
So as we saw, it's not a continuum, but the vesicles start bursting open and releasing their content at different parts. So it just goes from the point of entrance to the opposite end of that egg. So the presence of calcium seems to play a very important role in activating the development of the embryo. And here we have a little bit of a summary of it. So we have the sperm binds with the egg cell membrane. We have an influx of sodium that would allow for the rapid, fast block of polyspermy, because we have a change in potential and then we don't have any more sperm that gets attached. Also, that binding had a kinase stimulation that activates the phospholipase C, which is going to create product of the inositol phosphate that's going to help with the release of calcium. And then we know that this will have a different type of effects. So we have a degradation of the cyclin that will restore the mitotic cell cycle. Remember that all these eggs are being arrested prior to fertilization, right? We also gain the activity of NAD kinase that allows for membrane biosynthesis, but also calcium allows for the cortical granule exocytosis. That is more of the slow polyspermy block and the formation of the hyaline layer that will hold into these blastomeres once segmentation will start. At the same time, this deacylglycerol production activates the protein kinase C that have an exchange in pumps. In here, sodium and hydrogen will increase the pH, and that will put back into the cycle that egg. So now it's completely activated. DNA replication can start, and then cytoplasmatic movements gonna start being put in place so the egg can now become a zygote. Is this clear? This is schema here? Yep, okay.
Excuse me, Professor.
Yes, Maher.
What is the word PLC stands for again?
PLC, phospholipase C.
Okay, fine. Thank you.
Okay. Okay, so delayed response. Delayed response is that intracellular pH increase that I mentioned right here. Okay, so that is delayed response. So we have an increase in the influx of sodium ions, which causes an efflux of hydrogen. Remember that we need to balance, and we cannot have too many positive either inside. So one goes in, something goes out. And normally, in this case, is the hydrogen. So the pH increase along calcium ion increase turn on many different enzymes that will stimulate DNA synthesis. So here we have a little schematic of what happens. This is our fertilization. So very soon during fertilization, and this is the ratio for protein production. So right after fertilization, we see an increase in activity that coincides normally with the amount of calcium and sodium, that is going inside that cell, and the efflux of hydrogen, of course. So the wave of free calcium ions will normally inactivate what we call the mitogen activated protein kinase, and that is the MAP kinase. Okay, by inactivating this mitogen activated protein kinase, DNA synthesis can resume. Okay, so inactivation of the MAP kinase causes an activation of the DNA synthesis. Okay, so here we have the percent of ribosomes in polymers, and we see that they just go high. So we have more ribosomes activated and within hours of development, which means that we have a synthesis going on. And fertilization going to be using the RNA messenger that is stored in the ooplasm to start producing the cell divisions. Okay. So now we just have the sperm that entered the egg, triggers a whole whack of reactions to get that egg awake. That's like the keys of the prince to the how you call that, the Sleepy Beauty. That's it. All it comes in Spanish, but that doesn't come in English. I have never heard these little stories of princess as in English. So my boys, I read all the little stories in Spanish. So I never read these things in English. So the sperm gives a little kiss to the Sleepy Beauty just to wake up. Okay, come on, let's do something, right? So now that's part of the reaction for that egg, but now we need to meet them together. So they know that they are in the same place, but now they need to join together. So here is the fusion of this genetic material. And after fusion of the sperm and the egg, the sperm nucleus and its centriole separate from the mitochondria and the flagellum. Okay. So normally the mitochondria and the flagellum will be degraded. The egg comes from a female, and we are very territorial, I should say. I don't know about you, but I think all females are very territorial. So we don't want too much of a foreign thing, things that don't need to be there, right? So they're going to destroy that flagellum. And they're going to destroy that mitochondria. That's what the inherent material for mitochondria comes from the mother, because there is a mechanism for them to destroy anything that was in that mid piece of, remember, the mid piece of the sperm is packed with mitochondria because it's what provides the energy for the flagellum to move and the sperm to advance. Okay? So the flagellum and the mitochondria want to be disintegrated because the female egg contains many proteases that are going to destroy wherever it doesn't belong there. Okay. So there are few, if any, sperm-derived mitochondria are found in the adult organism. It's very, very rare that that happens because most of the mitochondria that is inherited comes from the mother. Okay. The centrosome needs to produce the mitotic spindle, and it comes from the sperm. It doesn't come from the mother, it comes from the male. Now, mice is an exception to this rule. Okay. For the rest of the species, it comes from the sperm, that mitotic spindle. The haploid egg nucleus at its stage is what we call the pronucleus. So it's not called nucleus. Before they fuse, it's called the pronucleus. So the haploid egg is the female pronucleus. Okay. And once inside the egg, the sperm nucleus want to start decondensing. Recall that the sperm nucleus is highly condensed, right? We saw it in the images before. And it will come decondensed, and it's called the male pronucleus. So we have a male and female pronucleus prior to fusion. This happens normally by the removal of the sperm-specific histones. We know that histone is what maintains these chromosomes really condensed and stored in the cytoplasm. So the sperm centriole forms the aster by forming microtubules, will be integrated with the egg microtubule. So both of them will be integrated, but it comes from the sperm centriole. Okay. Once the aster is formed, the two pronuclei will migrate to one another on the microtubules that have been established, and then they are capable to fuse. The fusion produces the zygote, which is diploid. Okay? And this entire process of entering, putting the centrioles that divide, putting that microtubules for the two pronuclei to migrate takes about an hour. So it's not too long for this to occur. Okay, so this is what we're going to see in this image. These are the nuclear events in the fertilization of the sea urchins. So here we have the sperm pronucleus that had decondensed, and we see the asters. You see this little, I'm just going to put it. Okay, let's go on. This was trying to do the, you see these little lines here, right? Those are the asters. Okay, holding on. So this is a sequential photograph showing the migration of the egg and sperm pronucleus towards each other in the egg. So this is the egg pronucleus, this is the male, and this is the time in seconds. So migration, migration, migration until they had fused. This is the sperm pronucleus, so tiny compared to the egg pronucleus that is very big. Okay, so we see these asters in the different color here. The macrotubules are colored in green, and we see that mainly comes from the male aster that start just going so rapidly and developing. Okay, so it is difficult to observe how a sperm and egg in mammals interact during fertilization in real time, unless it's in a petri dish, right? But in a petri dish, the environment is different. So there are so many factors that are not natural in this case. So mammalian fertilization is internal and normally arrives at the level of the oviduct. Okay. A sperm population ejaculated in the female is heterogeneous, containing spermatozoa of different stages of maturation. If we take humans, for instance, we have that a human can ejaculate something around 280 million sperm in just one ejaculation, okay? But this is what we call the ruffle theory, because they produce that many sperm in one ejaculation, 280 million sperm, but only 200 of them gonna get close to the egg. So they are so tiny, there is a huge universe in which they have been released and only few will get. So the numbers make the reason why at least producing many, some of them will have the chance to get close to that egg and only one will be able to fertilize, okay? So recent research indicates that it's more than just one way by which mammalian sperm can undergo the ultrasound reaction and bind to the zona pellucida. So in here we have the schematic of the mammalian fertilization tract. So we have here the uterus of the female, we have the fallopian trunks, we have the ovary, and this is the uterus, and this is the cervix, and this is where this thing's gonna come. So the egg of the sperm will use different mechanisms to arrive to the ampulla. So the ampulla is about here, and that is where normally fertilization will occur. If fertilization occurs right here at the level of the fallopian, at the entrance, very entrance, we're going to have an ectopic pregnancy, which normally is not going to arrive to a term, okay? So it takes about a couple of days for the oocyte to come from here and get into the uterus. So if it happens right here, normally it's going to niche about here, which will become an ectopic pregnancy. So, but in the ampulla, that's where fertilization needs to occur at this level, because then the membrane gonna still keep the integrity and only gonna get out of the membrane once it gets into the uterus to be able to establish the link with the mother. Okay, so the sperm takes around 30 minutes to go from the vagina to the oviduct, and this is mainly due to the spasms that arrive in the wall of the uterus of the female after a coitus. Okay. And those contractions are help. That's why when people are trying to conceive that had some issues with fertilizations, once the intercourse have done, normally females going to stay in the bed, put the legs up so sperm doesn't come down, and try to favor the fact that the sperm can still go at least to the roof here and then go to left and right, because those are the only two ways they can go. Okay, well, they can go out. Oops. So remember that a sperm is not necessarily active once it is ejaculated. Remember, I told you that last week. For some, you're going to take some of the vaginal liquid to be activated. For some, it's just water in external fertilization for some species. But the sperm will gain activity once it gets really close to the egg. What it's going to do is going to follow a temperature gradient that orientates. And by chemotaxis, we talked about this last week, all those chemical cues from the oocyte that are found in the cumulus of the egg, those are what is going to guide the sperm to where that trophy is, that is the egg. So contents of female reproductive tract capacitate and attract and activate the sperm. So at the beginning, what is it is released? It is very sluggish. It doesn't move too much. It just moves the tail a little bit. But as time passes by, within those 30 minutes, you're going to see the tails start moving and moving and moving and moving until it starts swimming really well. For males, we also have that the glare, that is the little mucus layer that comes out, right? Normally during the ovulation period, that is the optimal point for fertilization, is very, very slimy. It's very liquidy. Has a consistency, but it's still very liquidy. And that is where this sperm need to swim to go up. Okay. So this figure shows the hypothetical model for mammalian sperm capacitation. And unfortunately, my link to see this video here doesn't work anymore. But just trust me when I say that it starts moving really fast, the tail. Okay. So capacitation is a functional maturation of the spermatozoa. So the first step that we have is changes that take place via the sperm cell membrane, in which it may be that receptors are made available through the removal of cholesterol. So here we have the cholesterol in green attached, and here we have the egg albumin protein. So we're going to have a little bit of the receptors, okay, that are removed of cholesterol from the sperm cell membrane. This section here, the efflux of cholesterol from the sperm membrane, will allow the influx of bicarbonate that we have in here. And this bicarbonate, but at the same time, we have an influx of calcium. Okay, so bicarbonate and calcium will go in. These two ions, bicarbonate and calcium, will activate something that we call the SASI. The SASI simply is an activation of adelinate kinase. Adelinate kinase, excuse me. This activation of adelinate kinase causes an elevation of the CAMP, the cyclic AMP. High levels of cyclic AMP activates the protein kinase A, which is here. And at the same time, active PKA can then phosphorylate several tyrosine kinase that leads to the activation of the sperm. So it's a huge cascade, right? Just to say that is just not a matter of the sperm being released, that the sperm can fertilize an egg. It needs a whole cascade of activation. And once again here, see how calcium plays an important role in the cascade of activation. So not only calcium is essential for the activation of the egg metabolism, but it plays a very important role in the activation of the sperm, the capacitation of the sperm. So through the membrane alterations that happen, this is all in the sperm, right? The motility properties of the spermatozoa also change. So we're going to start discharging whipping movements of the tail together with a large sideways movement. So it is, I like my sperm, the sperm of the fish when it starts swimming. It just move the little head side to side, and then the tail starts whipping really hard. So once the sperm start moving, you're going to reach something that is called the hyperactivation, and that's what we're going to be seeing in the next slide, I think. So it reaches a state that we call hyperactivity level, and one can therefore say that is visible consequences of the capacitation. So if the spermatozoa is not capacitated, it won't be able to fertilize that egg.
Hi, I'm Dr. Alexander Travis, and my studies of sperm capacitation laid the groundwork for the CAP-SCORE sperm function test. Capacitation is a process necessary for a sperm to fertilize an egg, the first step in creating a baby. As they move up the female reproductive tract, sperm must respond to different stimuli that give them the ability to fertilize. As part of this process, specific components are removed from the membranes of the sperm. During capacitation, both the tail and head of the sperm undergo important changes. The tail takes on a hyperactivated pattern of swimming. The membranes of the head become more fluid, allowing key proteins and lipids to move into position for fertilization. These changes allow the sperm to move up the fallopian tube and through the difficult-to-cross, jelly-like matrix in between the cumula cells that surround the egg. Capacitated sperm are able to penetrate between the cumula cells. One of these sperm will make it through the zona pellucida, the thick protein coating around the egg. Only sperm that have capacitated are able to fertilize the egg.
So you saw how the tail start whipping a little harder. That gives them the speed, but at the same time, enough strength to penetrate the cumulus of the human mammalian beings. Okay, so this is very similar to what we saw with the sea urchin. Okay, this is in the mouse. So we have the sperm with the acrosome vesicle on top, we have the nucleus, we have the midpiece with the mitochondria, and we have the flagellum. Now, in sea urchin, we have the jelly layer, right? There was right here. And then we have the cell membrane. And then we have the egg membrane. In humans, in mammals, we have the cumulus layer, which is very thick, as we see here. And then we have the zona pellucida before we get to that egg membrane. Now, here we have the acrosome intact, reaches and binds to the zona pellucida, which is thicker than the vitelline envelope in the sea urchin. Okay. Then we have the acrosomal reaction only can occur and should occur when it binds to the zona pellucida, as we see here. Okay. Then the sperm will digest the zona pellucida. That's where the acrosomal reaction will occur. Okay, so we're gonna those enzymes, the proteases, etc., are gonna degrade that zona pellucida, and then the fertilization cone will be dig through the activation of those proteases. Okay, so open the hole until it binds to the egg membrane here. Now, if you see here, even though it approaches the zona pellucida in a perpendicular fashion, as we see here, as it gets through that zona pellucida, it starts tilting in an angle. Okay, and we see in here that the sperm and the egg membrane will fuse, but in more angle side. Okay, the recognition and how this binds. So the textbook doesn't tell us how a sperm penetrates the cumulus. Mammal sperm don't have the acrosomal process as we saw in sea urchin, right? You notice that? We don't have an acrosomal process here. Okay, just wait. It's just the acrosomal reaction that occurs, but we don't have a process. That's a little mini key that came out, remember, with the actin and binding exposed to the tip. So the zona pellucida of mammals play a role that is analogous to what the vitelline envelope in the invertebrate sea urchin we saw. So the glycoprotein matrix secreted by the growing oocyte plays two major roles during fertilization. The first one is that it binds the sperm, and the second one is that after the sperm is bound, it initiates the acrosomal reaction. Because mammals have internal fertilization, species-specific sperm recognition does not seem as important as in individuals that have external fertilization, where many other species can come, right? Hence, the binding of the sperm to the zona is relatively and not absolute species-specific, okay? The mouse zona pellucida is made of more or less three, although later papers have mentioned a little bit more, but normally we're going to have three major glycoproteins in the zona pellucida. We have the ZP1, ZP3, and ZP3. Simply is zona protein, ZP, okay? So three different zona pellucida proteins that are like the key and the lock in this system. So for the longest time, we thought that the glycoprotein zona pellucida 3 initially bind the sperm and induce the acrosomal reaction after the sperm have bound to it. But new research lately have demonstrated that most sperm that bind to the zona have already undergone the acrosomal reaction. So it is not obvious that most sperm undergo acrosomal reaction in the cumulus. Those that underwent into acrosomal reaction in the zona are unsuccessful. So they really need to wait until they get to the zona pellucida, right? Not in the part of the cumulus. So most sperm bind through the zona pellucida 2. That's what we know right now. That may change. This is not zona pellucida 3 protein, but zona pellucida 2. And in particular, it seems to be true, at least in humans. So let's see how this fuses in this section. So in mammals, the sperm contact the egg, not in the tip, as I mentioned, but more on the equatorial side, as we see in this image, okay? So that's what we call the equatorial region. So this right piece right here. So it still have the nucleus that is fully compacted. We have the acrosomal vesicle. In here we have our proteins, except that in this case it's not binding as we saw in sea urchin. In this in mammals we call izumo. Okay, so the acrosome reaction in addition to expelling the exzymatic content of the acrosome that would allow it to dig a hole and come across that zona pellucida into binding to the cell membrane, it have also all these parts. So we don't have an acrosome as we have in the other ones, but we have this section here. So we have a degradation of the membrane. Remember the video? The person said that we're going to start removing stuff from the membrane to allow this reaction. Okay. So the junction between the inner acrosomal membrane and the sperm cell membrane is what we call that equatorial region, and is where the membrane fusion between the sperm and the egg begins. So we have it here. Here we see the membrane start disappearing. That's the sumo, that's the acrosome proteins in green. And here we have the microvilli of the egg with the specific receptors, and the specific receptor is called the Juno CD9. Okay, so start placing into the equatorial zone until it binds, and then start digging the hole to get into fusion of the cell membranes. Now, same aspect as we saw in sea urchin, we need to try to avoid polyspermy. We know the consequences. So have a similar slow blocked reaction as we saw in sea urchin, but in mammals, the cortical granule reaction does not create a fertilization envelope. So that's one of the main points. But the ultimate effect is about the same, okay? So we're going to have a release of enzymes that modify the zona pellucida, the sperm receptors, and then they can no longer bind to more sperm, okay? This process is what we call the zona reaction, and involves modification of the zona pellucida to that we know that is mainly the receptor to which the sperm are going to bind, okay? So normally what happens is that the zona pellucida 2 is clipped by the cortical granules by a protease that is called ovastacin, okay? And then the zona pellucida will lose its ability to bind to the sperm, okay? So once the sperm has entered the egg, other sperm can no longer initiate or maintain their binding to the zona pellucida and are rapidly shed off the surface. In mammals, the fusion of the genetic material between the male and female pronucleu normally takes about 12 hours. So it's a little longer than in the sea version that we say just merely an hour. In here, it takes about 12 hours. So when the sperm enters the egg nucleus is arrested in metaphase 2 of meiosis. That's what we saw last week, okay? So unlike the version egg, which is already haploid, mammalian oocyte still has chromosomes in meiosis, okay? So DNA of the sperm nucleus is tightly bound with the proteins that we call protamines in here. In the egg cytoplasm, the disulfide bonds of the protamines are broken, and these are broken by the intervention of glutathione, and the sperm chromatin start uncoiling, start diffusing, okay? And then the male pronucleus start enlarging. Calcium oscillations, saw it before, in this case it will be doing the same. Calcium oscillations activate a kinase that causes the egg to go back into the cell cycle. That's exactly what we saw also in sea urchin, okay? So the oocyte nucleus does complete its meiosis 2. The centrosome from the sperm using proteins stored in the ovum, because remember, the sperm doesn't have too many stuff inside, right? It has the nucleus and just a few mitochondria in the midpiece. So the centrosome of the sperm will use some proteins that are stored in the ovum to make the aster. Okay, and then the microfilaments of the aster contact the female pronucleus. The pronuclei start migrating to one another, as we saw in the previous with the sea urchin. Okay, and then they're going to start approaching to one another. When the two pronuclei meet, their nuclear membranes will break down, but they do not form a common zygote nucleus, as we saw in the sea urchin. Okay. Instead, each cell chromatin condenses into chromosomes that attach to the spindle fibers at the metaphase plate, such that the chromosomes from the sperm and the egg intermix on that metaphase plate. So if we compare the mammal embryo or early stage, the true zygote nucleus in the single cell stage never forms. A true diploid nucleus in mammals is first seen not in the zygote, but in the two-cell stage. So each sperm binds into the egg, not only its pronucleus, but also the mitochondria and the centriole, and a little bit of the cytoplasma goes in. The sperm mitochondria and their DNA are degraded in the cytoplasm, the same thing as we saw in the sea urchin. Okay, and this is by dilution and by actively targeting them to destroy them. Now, I think it was last year or 2 years ago that we saw that some babies have the male mitochondria. So even though we know that this happened, there are some mitochondria that can bypass the system. So we can have some heritable mitochondria coming from the male. Okay. But the centriole normally is maintained to help with the creation of the mitotic spindle. So the centriole here is not destroyed, it's kept for the mitotic spindle formation. Okay. So in resume, this is what we can see in this figure. So this is the human fertilization. Microtubules are stained in green, as we see all around here. And the DNA is colored in blue right there. So here in A, where is my mouse? Right here. Okay, so here in A, mature unfertilized eggs that complete meiosis 1 and produce one polar body, which is seen right here. That's the first polar body. Okay. In B, we have that the sperm got inside the egg. This is right here inside, here on the left. Okay. Microtubule condenses. So this is what you can see, all the microtubules. Okay. And at this point, the egg, because it just got fertilized, completes meiosis 2. Okay. About 15 hours after fertilization, we can see the two pronuclei, 1 and 2. And here what we see right here is the tail of the sperm. So we know that this is the male pronuclei right there. Okay, so the new pronuclei start approaching to one another. This is what we see right here. The two pronuclei are close together. And the centrosome splits to organize the bipolar microtubule array. The tail is still intact. It hasn't been completely destroyed. Okay, it's right there. In D, in this one here, a mitotic spindle appears. Okay, so this is the mitotic in prophase. We see it right there. The sperm derived and the egg derived chromosomes condense separately. We don't see it too well, but they do. Okay, and we still see a little bit of the tail right there that continues to disintegrate. Okay, and then in E, what we have in this image here is that the chromosomes form sperm and egg intermix on metaphase plate, and we're going to start the mitotic spindle. So we're going to see in this little short video how the pronucleus are migrating. So this is the female and this is the male under here. Look at the size difference between the male and the female. The female is bigger than the male, and it goes really quickly in real time. So it approaches and then move together in the metaphase plate. So preparation for cleavage. So we have in mammals phospholipase C. There's the PLC zeta. This little wiggle here is the zeta. Okay, so PLC zeta from the sperm is responsible for egg activation and pronucleus formation. We didn't saw that necessarily in the sea urchin, right? In the sea urchin was the egg. In sea urchin, eggs are activated as a single wave of calcium from the entrance point of the sperm. In mammals, numerous waves of calcium traverse the egg. So that's a big difference in here. We have different times. It's too long, right? So it takes about 12 hours for this to happen. So it requires multiple waves of calcium to be able to do that job. So the multiple duration, the amplitude and duration of the number of calcium waves regulate the timing of mammalian egg activation. So calcium released by inositol triphosphate, the IP3, the one that we have in here, binds in this case to calmodulin, okay, activated protein kinase, which eliminate the inhibitors of the RNA translation, and it binds to the mitogen activity, activates protein kinase, the MAP that we have seen before. So, oops, click wrong. So we can have meiosis resumption, okay? And also it allows the synaptotagmin, which help initiate cortical granule fusion. This is a full mouth word. I just need to pronounce it so slowly so I can say it correctly. But that's what activates the granule, the cortical granule reaction. So if calcium influx, for instance, is blocked, the second polar body does not form. And then the result will be two non-viable triploid egg pronuclei. Remember, we need to get the right amount of chromosomes, right? So we go from 2N to 1N, so we can fuse together. Okay, so in mammals, fertilization includes contact and recognition between gametes. A sperm and egg are mutually activated, and both activations involve calcium ions and membrane fusion. Fusion of gamete is mediated by protein molecules that allow merging of membranes, like in binding. We have a fast and a slow blockage of polyspermy, so cortical granule reaction, pH change, and calcium increase, all to activate the egg to form the fertilization membrane. And calcium also mediates egg metabolism activation, protein synthesis, and DNA synthesis. Now, because we learn every day, and I say that most species have these two mechanisms to avoid polyspermy, we're going to see what happened in birds, because as I always say in my vertebrate physiology class, I always say it's also in this course, the world is full of exceptions, and exceptions are so cool and interesting.
So we're taught at school that fertilization is a process that involves one egg and one sperm. That's kind of the traditional way that we learn about it, but that's not always the case with birds. Can you tell me a little bit about how fertilization works with birds?
Yeah, so you're right, that is what we typically learn in school. And actually that is kind of right for all animals, including birds, because the actual process of fertilization, so the fusion of the male DNA from the sperm with the female DNA from the egg, is really a one-on-one affair. However, the events leading up to that the actual process of fertilization can vary quite a lot between different animal groups. So if we just take humans, for example, because that's what we're kind of familiar with, in the case of humans and other mammals, one sperm penetrates the egg, and then very quickly the egg forms a block to all the other sperm. And this is really important because in mammals, if more than one sperm enters the egg, and we call that process polyspermy, then this is really quite destructive to the egg. It can cause abnormal development of the embryo and ultimately lead to the embryo dying at a very, very early stage. But in birds, in contrast to mammals, tens, hundreds, even thousands of sperm can penetrate the egg. Wow. You still only get one of them fusing with the female genetic material, but there are several others right there in the vicinity of the female pronucleus, all kind of right next to the action. And we've kind of known this for, surprisingly, over a century. So even in the 1900s, a guy called Eugene Harper had seen all this stuff going on in a pigeon's egg, but no one had asked kind of what's going on with these extra sperm. Do they have any role to play in fertilization or embryo development? And so this is was a big kind of unanswered question.
So what does happen if only one sperm penetrates the ovum of a bird?
Well, our research has shown that in birds, kind of completely the opposite to mammals, one sperm really isn't enough. So if one sperm penetrates, it may well make its way to the germinal disc where the female pronucleus is, and it may well fuse with the female pronucleus, so fertilization can take place. But actually, that fertilized egg is very, very unlikely to develop beyond the very earliest stages of embryo development. So our experiments have shown that actually, in order for normal embryo development,
What's my fault?
can cause abnormal development of the embryo and ultimately lead to the embryo dying. Penetrate the egg. Wow. You still only get one of them fusing with the female genetic material, but there are several others right there in the vicinity of the female pronucleus, all kind of right next to the action. And we've kind of known this for, surprisingly, over a century. So even in the 1900s, a guy called Eugene Harper had seen all this stuff going on in a pigeon's egg, but no one had asked kind of what's going on with these extra sperm. Do they have any role to play in fertilization or embryo development? And so this is was the big kind of unanswered question.
So what does happen if only one sperm penetrates the ovum of a bird?
Well, our research has shown that in birds, completely the opposite to mammals, one sperm really isn't enough. So if one sperm penetrates, it may well make its way to the germinal disc where the female pronucleus is, and it may well fuse with the female pronucleus, so fertilization can take place. But actually, that fertilized egg is very, very unlikely to develop beyond the very earliest stages of embryo development. So our experiments have shown that actually, in order for normal embryo development to proceed, you actually need multiple sperm penetrating the egg of a bird.
Okay. And your experiments, you just mentioned, are kind of investigating this and looking at the difference between sperm depleted males and kind of a control group. So can you just talk me through exactly what you did in your experiments?
Yeah, so we actually looked at this in two very different bird species. So we looked at it in a little passerine bird called the zebrafinch, which is really tiny. It actually lays eggs which are just this big.
Absolutely tiny.
And then the other species that we looked at was the domestic chicken, which I'm sure you're familiar with. And actually, we started off with the zebrafinch, and we kind of started this study a little bit by accident, to be honest, because what we were trying to do was establish a protocol for doing artificial insemination in zebrafinches. But as I said, they're so tiny that actually it was really difficult, and it didn't really work. So what we found is when we tried to artificially inseminate zebrafinches, almost no sperm made it to the egg. Often no sperm did make it. If they did make it, they were in very tiny numbers, like just less than 5, sometimes even just 1. So by accident, we'd kind of found this way of severely limiting sperm numbers reaching the egg in zebrafinches. We still don't know exactly why that was the case, but nonetheless, we kind of had this method. So we decided to look at, compare whether females that had very few sperm reaching the egg, whether there was any differences in terms of their fertilization success and embryo survival, compared with what we called a control group, but what was actually females that had natural copulations with males. Of course, this wasn't the ideal comparison because there's lots of other things that could differ between artificial insemination and natural insemination. So we actually then decided, okay, we actually need to try and do this properly with a real controlled experiment. So we repeated the whole thing on domestic chickens, in which artificial insemination is well established. So we could do a very low sperm dose and a very high sperm dose, both artificially.
And what did you find was the difference between the two groups in terms of fertilization success and embryo survival?
So in terms of fertilization success, there actually wasn't a great deal of difference, and that was quite surprising.
So such a big difference in sperm numbers.
Yeah, exactly. So in the zebrafinch, whether eggs were penetrated by 1 sperm or say 50 sperm, there really wasn't any difference. They were just as likely to be fertilized. In the chickens, there was a 20% reduction in fertilization success in the sperm limited group. So a kind of quite a decrease, but a small decrease. But funny enough, there was quite a strong pattern there. So basically, those eggs that weren't fertilized had all been penetrated by fewer than 3 sperm. Whereas the ones that had all been penetrated by 3 or more sperm. So it does look like there's some kind of threshold effect there. We'd have to do a bit more work to be able to actually say that 3 is kind of the magic number of sperm for chickens, but it does suggest that there might be kind of a threshold number. In terms of embryo survival, though, there was a real striking difference. So only 11% of the embryos that were produced under sperm limitation in the zebrafinches actually developed to hatch. And so in real numbers, that's just 3 out of 27 embryos.
Oh, that doesn't sound like much.
No, exactly. And it was even, the effect was even stronger in the chicken. So 10 embryos were produced under sperm limitation, but none of them hatched. In fact, none of them even survived beyond 48 hours of development. So there really does seem to be a real strong effect of the number of sperm penetrating on subsequent embryo development and survival.
That's really interesting.
Okay, so just want to put this back into... Oh, I need to stop sharing my screen. So it is really interesting. I find that it's really interesting to see that even though for most of the species we say only 1 sperm should enter, in birds it varies, and the amount required, it also varies among the species. And it's not as a matter, as I say, that the male and female pronucleus join and fuse together. Is what is the effect of them in the survival of the development of that embryo. So more than 3 seems to be enough to force that fusion, but the survival of the embryo is being affected too. So I always like these examples of things that are not necessarily as we thought they should be, but hey, just a little short comment, because I know that I have received a couple of emails looking at this. The outline that is due on Friday it has a couple of things. One is just to be sure that you have joint contact discussed with your partner, okay? So that's what I have, that deadlines. Okay, just meet with the partner so you can start working on that. Both of you are working together towards creating this, and it's not just one person. If you, as I say, if it ever happens, just let me know. Don't wait until the very last second to say, well, I did it all because my partner didn't join me. So you both have responsibility to work on this. This work is per review too, so you need to tell me. You're gonna receive a form at the end just to let me know if you and your partner work together, and you gotta be honest. Okay, it's not because it's your friend. If your friend is your friend but didn't help at all with anything, does it deserve to have that percentage on you because you did the homework? No, right? So I trust you that you're gonna be totally honest. An outline is simply just to see that you also started a research, right? So I need some subtitles of what you think right now will be included into your video. Okay, so a definition of the condition. This is to be submitted into the Dropbox that is already in the Dropbox, and it says video work assignment. Okay, so a definition, prognosis, diagnosis, just a little bit of subtitles. I want just to see that you have already started, okay? I know that we have this tendency, I have enough time, and then it's like, oh, fuck, it's tomorrow, we need to submit this, and then it's panic, and I want you to give me the best that you can do. So this is what I have put some deadlines. As I always have said in the past, if you finish your video before the time and you want me to go over to guide you to say, oh, your voice is too half, it's too quick, too much images, the images are not clear, I think that you need to focus a little bit more into this or into that, please do so. It's my pleasure to help you. I want you to do well, okay? But if you leave it to the very last minute, if you send it to me the night before the date, that just forget it, I won't do it, right? If you didn't rush what I need to rush now to give you feedback, forget it. Okay, so it's very simple, 1 page, that's all, the name of the 2 people on your team. I have a list of you guys, and just a couple of lines saying the definition of the condition is this, I'm gonna do this and this. It's just an outline, just some points in there just to show me that you have already started working on it. Is that clear? Nothing complicated. The abstract, the 500-word resume, is only to be submitted on the second, whatever is the date that I say that you are submitting the video. And all of these 3 things, the outline, the video, and the abstract, need to be submitted, all 3 in the same Dropbox, okay? So only one person needs to submit, per team needs to submit the work, okay? I don't want to have each of you submitting. Just say one person is going to submit the work, okay? And just be sure that that person has done so, okay? I don't want to have 60 of the same, which means 30 of the same because you are working in pairs, okay? So Dropbox and that. Remember that the video has to be saved in MP3 format, okay? Because it's easier. The outline is due on the 29th, yes. That is what it says in the Dropbox, and that's what it says in the syllabus, and that's what I have said last week. So on the 29th, there is a time too. I can't remember, it's 4 p.m. or something like that. I can't remember. It's to be submitted on the Friday. Okay. Friday, we're going to have article discussion, and we are going to have a lab. So I released the notes on Friday. You should have started working. If not, you have this week until Friday to work on that. If you have questions, that is when we're going to meet in the lab to respond to any of the questions possible that you may have. You got to use the images that I gave you, nothing else, okay? And the lab report is to be submitted on the Saturday, just in case you needed an extra day, but you had all the notes on the Friday. So this Friday, I'm going to release the notes for the following lab, okay? So you have a full week to do this. Do you have any questions? Is that clear?
Yes, Professor, regarding the lab 1, for the small answer questions, do we have to write the reference or only for the long answers?
No, everything has to be referenced.
Okay.
Yeah.
So we can reference from the Planarian article that you have posted as well as
yes,
other...
Absolutely. That's what I posted that paper along ahead.
Yeah, but we can use other references as well, right?
Oh, absolutely, absolutely. You can reference wherever, but you need to reference. This is a 4th year level course, so I'm expecting you to comply with how we reference. So every single sentence, as I say, has to have a reference because there is no such a thing as common knowledge. Everything that we know came from somewhere.
So you're also looking for in-text citations?
Exactly.
Okay.
And full sentences that yes, no is not an answer. That's the answer that I maybe, and not even first-year students. So when I say explain, it's full explain. And I put the points so you have an idea of how in-depth you gotta answer, okay?
Also, another question. In the outline, are you looking for it to be as just a blank text as a paragraph or 3, or in form of points? So slide 1 is about this, slide 2 is about
point form from the outline is okay.
Sorry?
Point form for the outline is okay.
Okay.
As far, and remember, the APA format, yes. And in the syllabus, I even give the link to go and check how the APA format is. What I was about to say, I forgot. What happened when my brain sees something else. Any more questions? Yeah, a YouTube video, if you are using one, yes, everything needs to cite. And the link that I put in the syllabus into how to cite indicates how to cite web pages. Any more questions? Elena, yes, the sperm gets aroused. Yes.